Hello friends, today I am going to just discuss about the antibiotic sensitivity test. The antibiotic sensitivity test is also known as antimicrobial sensitivity test or ABST. Okay, so why the ABST is to be conducted? I am going to tell about the detailed procedure of the ABST. So first of all, we have to take sample. The sample may be either milk, either blood, either any kind of pus, okay, urine. So there may be any kind of sample like nasal discharge. So it should be collected in and immediately is to be takes place into the 4 degree centigrade in the refrigerator or to be transported in chilled material. Okay, so the sample is to be collected either by swab or either as a whole sample is to be collected, then is to be transported in refrigerator or in a chilled at 4 degree centigrade temperature. So in the microbiology department or in the laboratory, we have to inoculate into the BHI broth. Okay, the full form of BHI is the brain heart infusion broth. Why we are telling the brain heart infusion broth? Because this broth contains the extract of the brain and heart. And why this infusion broth is to be taken? Because it is rich in the nutrients and it is almost richer than the normal nutrient broth. So the BHI is to be taken just for the enrichment. It is an enrichment broth, and after that inoculation into the BHI, we have to put it at 37 degrees centigrade for 3 to 4 hours for the incubation. Why the incubation is required for 3 to 4 hours only? Because the bacteria tend to log phase at this time or achieve the log phase at during this time, or the bacterial growth or bacterial division is very high during this time. So the log phase of the bacteria is required for our ABST test. So after incubation to 3 to 4 hours, there may be the turbidity is to be seen in the broth. Okay, so there may be the turbidity is to be seen. So the turbidity is to be standardized by using McFarland tube. Okay, so I'm going to tell about what is the McFarland. So there is a McFarland standard. You can also search on the internet the what is the McFarland standard. So I am not going to brief during this time. I will cover it in another lecture. So we have to standardize the our turbidity in the broth by using McFarland standard, and we have to adjust the McFarland standard 0.5 level, and by adding the broth or the adding BHI broth into the tube to adjust the turbidity of the bacteria to just a 0.5 standard of McFarland. So after standardization of the turbidity by the McFarland standard method, we have to inoculate the sample, the turbid BHI broth, the loop full of broth or the 2 to 3 drops of the broth onto the Muller-Hinton agar plates. So why the Muller-Hinton agar plates is to be taken? Because it is the best studied agar for the antibiotic sensitivity test. So either the Muller-Hinton agar should be sterilized, and the sterility check is to be done by keeping these plates after preparation into the incubator for at least 12 hours, and after sterility check we have to use the sterile Muller-Hinton agar plates. So by swabbing of the turbidity onto the Muller-Hinton agar plates, we have to gently swab the whole plate by using this turbid broth. So we have to plate the Muller-Hinton agar plate by swabbing, and on that swabbed Muller-Hinton agar, we put the antibiotics disc. Okay, so normally in our lab we put about 8 disc on that plate for checking the best antibiotic resistance. So the disc is to be keep uh, on the agar gently, and further we will inoculate this plates uh, for 37 degrees centigrade for 24 hours. So after 24 hours, what we have to do, we have to just check the zone of inhibition that is created by each disc. The inhibited area is just because of the unable to grow the bacteria at the zone of inhibition site. So the zone of inhibition is to be checked and is to be measured. So you have to keep in mind that the disc is about 6 mm in the size, and the zone of inhibition is to be measured from the periphery of one circle to the another periphery of circle. If the circle is not like round shape, and it is like a dome shape, so we have to measure the end of one circle from this point to the another end of the circle, and we should calculate the circle, and we have to check the table that is supplied by the supplier with the antibiotic disc, and this table is to be said that the standardized already standardized by the supplier, and further we can decide either this is the resistant or either this is a sensitive antibiotic for the microbes. So this whole method is based on the Bias et al 1966, and this is also called as disc diffusion method. So this is a method what we are calling is a disc diffusion method. So here we should know that the swab should also be sterilized during swabbing on the Muller-Hinton agar. Okay, so this is all about the ABST, or the antibiotic sensitivity test, and it is a very common routine test to measure the resistance of the bacteria to a particular antibiotic in the field, also in the diagnostic labs. So thanks for watching this video. Thanks.